Hello guys, my name is Connor Allen. I'm going to be talking to you guys about the cell cycle. So first, the learning objectives. You're going to know how the cell cycle works. You're going to know the phases of the cell cycle, understand different stages inside the phases of the cycle, learning about mitosis and its functions, animal and plant cells, and the duration of the cell cycle, and much more. So first, let's talk about what is a cell cycle. A cell cycle is a series of events that take place in a cell as it grows and divides. The cell cycle is the name we give the process through which cells replicate and make two new cells. The cell cycle has different stages called G1, S, G2, and M. And on the right, if you're a little bit confused, I didn't mention anything in the mitosis because we'll get into that later. First, interphase. This is the longest phase. This is the longest part of the cell cycle. The cell undergoes normal growth processes while also preparing for cell division. In order for a cell to move from interphase into the mitotic phase, many internal and conditions must be met. So there are three stages of interphase, which are called G1, S, and G2. And you see on the right in the picture, there are some objects, or there's some objects like centrioles, chromatin, nucleolus, and you'll understand a lot about that later and how they work. So first, G1 phase, or called the first gap. The first stage is called G1 from a microscopic, and because from a microscopic aspect, little change is visible. However, during the G1 stage, the cell is quite active at the biochemical level we're just not able to see under the microscope. The cell grows and accumulates the building blocks of chromosomal DNA and the associated proteins, as well as sufficient energy reserves to complete the task of replicating each chromosome in the nucleus. Next, talk about the S phase or synthesis of DNA. So this is the longest stage of interphase because of the complexity of the genetic material being duplicated. DNA replication results in the formation of identical pairs of DNA molecules, sister chromatids, that are also firmly attached to the centromeric region. Centrosomes are also being duplicated. Two centrosomes will give rise to the mitotic spindle, that apparatus that orchestrates the movement of chromosomes during mitosis. Centrosomes are associated with centrioles, and centrioles help organize cell division. Next, talk about the G2 phase, or the second gap. The cell replenishes its energy storage and synthesizes proteins necessary for chromosome manipulation. Some cell organelles are duplicated, and the cytoskeleton is dismantled to provide resources for the mitotic phase. There may be additional cell growth during G2 as well. The final preparations for the mitotic phase must be completed before the cell is able to enter its first stage of mitosis. Next, the M phase. During the mitotic phase, or the M phase, the cell divides its copied DNA and cytoplasm to make two new cells. This phase involves two distinct division-related processes, which are called mitosis and cytokinesis. Mitosis. This is when the nuclear DNA of the cell condenses into visible chromosomes and is pulled apart by the mitotic spindle, which is a specialized structure made out of microtubules. The mitosis takes place in four stages. Prophase, which is sometimes divided into early prophase and prometaphase, and there's metaphase, anaphase, and telophase. And on the right, there's just an image that shows how everything works. So the prophase, or in the metaphase, after the prophase, they form spindles. In anaphase, they're on two different sides. It opens, and then telophase, you can see it's forming into almost two new cells, and then at the end, that's how the two new cells come apart. So, phases of mitosis. Prophase, the first phase of mitosis. This is the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Next is metaphase, which chromosomes are at their second most condensed state in a line at the center of the cell. This stage lasts about 4% of the cell cycle's duration. Next, anaphase. This is the shortest phase of mitosis, the process that separates genetic material in a parent cell's nucleus into two identical daughter cells. By the end of anaphase, the two halves of the cell have the same collection of chromosomes. Finally is telophase. It's the final stage, and it occurs when replicated chromosomes have been separated and pulled to the opposite poles of the cell, and the nuclear membrane reforms around the chromatin, forming two new nuclei. Now, cytokinesis. So, the cytoplasm of the cell is split into two, making two new cells. This usually begins just as mitosis is ending, with a little overlap. But most importantly, cytokinesis takes place differently in animals and plant cells, and we'll dive into more about those two cells later. So, the G0 phase. I didn't put this in there because it's technically not a part of the cycle. It's only when it's known as the resting phase, so it's a non-proliferative state of the cell cycle where cells are either at rest or performing basic functions. It can last from a few hours to a few years, depending on the circumstances. So for example, reproductive cells have a very short resting phase, while plant seeds have been known to germinate many, many years after they have been deposited. Next, animal cells. So, in animal cells, cell division occurs when a band of cytoskeletal fibers, called the contractile ring, contracts inward and pinches a cell in two, which is a process called contractile cytokinesis. So, the indentation produced as the ring contracts inward is called the cleavage furrow. Animal cells can be pinched in two because they're relatively soft and squishy. So, you see in the diagram here, you see how the indentation in the middle from the cleavage and the contractile ring, it forms two new cells. So for plant cells, these are a little bit different. Plant cells are much stiffer around the animal cells. They're surrounded by a rigid cell wall and have a high internal pressure. And because of this, plant cells divide into two by building a new structure down the middle of the cell. This structure is known as the cell plate. The cell plate is made up of plasma membrane and cell wall components delivered in vesicles, and it partitions the cell into two. So you guys might be thinking, how long does a cell cycle take? So different cells take different lengths of time to complete the cell cycle. For example, a typical human cell might take about 24 hours to divide, but fast-cycling mammalian cells, like the ones that line the intestine, can complete a cycle every 9 to 10 hours when they're grown in culture. Different types of cells also split their time between cell phases in different ways. So, in early frog embryos, the cells spend almost no time in G1 and G2, and instead rapidly cycle between S and M phases, resulting in the division of one big cell, the zygote, and into many smaller cells. What else can play a role in cell division? Proteins. Proteins play a huge role in stimulating cell division and can be classified into four groups, which are the growth factors, growth factor receptors, signal transducers, and nuclear regulatory proteins, or also known as transcription factors. And on the right, that's just how the protein works. So let's dive into more about those growth factors. So for a stimulatory signal to reach a nucleus and turn it on, cell division, four main steps may occur. First, a growth factor must bind to its receptor on the cell membrane. Second, the receptor must become temporarily activated by this binding event. Third, this activation must stimulate a signal to be transmitted or transduced from the receptor at the cell surface to the nucleus within the cell. Finally, transcription factors within the nucleus must initiate the transcription of genes involved in cell proliferation. So there might be some terms that you're like, oh, what is this? So I'm going to talk to you guys about some of them. So transcription, the process by which DNA is converted to RNA. Chromosomes, these are known, but I'm going to go over them. So they are a thread-like structure of nucleic acids and proteins found in the nucleus of most living cells, which carry a genetic information in the form of genes. Chromatin. This is the material of which the chromosomes of organisms other than bacteria are composed, and it consists of RNA, DNA, and proteins. So centromeres. They are a membrane-free organelle that serve as microtubule organizing centers in distinct eukaryotic lineages. Now last, centrioles. Centrioles are a minute cylindrical organelle near the nucleus in animal cells, which occur in pairs and involved in the development of spindle fibers in cell division. These are my references, and thank you guys for listening. I hope you guys learned a lot and attained information about the cell cycle.